about DNA processing and samples life, and we will discuss sustainable models for genetic diagnostic companies, and we will look at existing companies in the world, and I want to share with you my view of ways of collaboration in running projects, and also the term of and the determination of intellectual property is very important nowadays in a new economic model. So protection of intellectual property is extremely important in nowadays business. And we'll talk about global availability of genetic tests and regulatory affairs. So, what is genetics and what do we obtain after genetic test? Okay. These, do you see it well? Maybe we should.
It's okay.
Light. These are lots of letters, ACGT, nucleotides, and this is the sequence, marked up sequence as showed in the ensemble database. And the red markup is exome. It is a part of a gene that finally is converted to the protein. And the white unmarked sequence is intron. And yes, much better.
Not better. Continue, don't worry.
Okay. So introns are not converted in protein. And there are different approaches in genetic testing. More detailed is whole exome sequencing, and whole genome sequencing is more detailed information. But all exomes of genes contain. What happened? I see. Let's make a break.
Oh, it'll be right now. It'll come up in about 15, 20 seconds.
Okay.
Yeah, the power's not.
So we have 3 billion pairs of these letters in our body. And these letters help us to construct the small briquettes of our body, proteins, from which we consist. And exome is the quantity of all exons from all genes. And it is about 1% of whole genome.
We trust.
Okay. It was an experiment with life. Let's turn it on, please.
It is on.
Are there experts in genetics here?
You're alone, I think.
You want an expert in projectors, not in genetics.
I just want to know what kind of details should I go through to make the material more easy to understand, because genetic testing is a multi-level stuff. Maybe it is switched off?
No, it's switched on.
Let's turn on all the lights.
No, that's not at all.
Нам сейчас технарь больше нужен, чем специалист по генетике. Все хорошо, очень хорошо. Молодец.
Начало положено.
Не, давай, в том же духе.
Да, пожалуй, это было.
Слушай, у нас прекрасная видеосъемка идет. Ты что, сразу в YouTube выкладываем? Как бывает на международных конференциях. Смотри, целых 2 профессора тебе пытаются помочь.
Может, сейчас все возникнет?
Я думаю, что да.
Значит,
Сейчас как раз ты 1,5 часа и растянем.
Я поняла, зачем.
Зачем столько времени?
Тайм-менеджмент имеет свои особенности.
Ну, раньше нельзя и позже тоже нельзя. Но сейчас он нагреется. О,
I'm very
perfect.
sorry. Thank you so much. Thank you a lot. So we have a puzzle of 3 billion pairs of letters, and we want to read it, to understand. And usually it is quite time constrained because we have a patient and the patient waits for our help. And we have to interpret data and to make conclusions quite quickly. So this is the DNA, and for last 50 years there were 3 levels of genetic information obtaining. The first level is a single nucleotide
Два профессора
polymorphisms.
пытаются поухаживать за нашей девушкой.
This
Таким
means
странным
that we
способом
get
помогают ей устанавливать.
several letters from the
Проекты.
genome by PCR technology. And if we know the disease and the pathology and pathogenesis of the disease in the body, we can use this technology very fully and thoroughly, and we can obtain an information that helps us make a clinical diagnosis. The second stage, the second level, is sequencing, the gene sequencing. The standard technique is Sanger sequencing. And in this technology, we obtain a gene sequence, which is marked as exon and excissored introns. And having this information, we may conclude more detailed information about the disease pathogenesis. And last 10-15 years, a new generation sequencing is being developed. And there are several companies working on developing of this technology. And one of the most popular and widespread is Illumina company. And to make you just observe what is it, the results of this sequencing. It is a text file of approximately 30 gigabytes, and it is short reads technology. So we obtain short reads, short letters that are arranged in a text file, and this text file is very heavy. And if Sanger sequencing is already known for 30-40 years, and we can interpret the results because the genes are not so long. So when we get next generation sequencing technique with having whole exome and whole genome, we obtain data which we cannot use as we did it in Sanger sequencing. So new techniques, new programmers and algorithms in bioinformatics is needed to use this information, because usually we try to find a pin in a haystack in the clinical case. So it is quite hard to obtain it. There are several types of programming algorithms, like BVA alignment, SNP effect annotation, mark variant calling, other filtering, but the most important thing is to integrate it with the obtained databases in medicine. And these databases are PubMed and Ensemble, working with gene and genomes, GeneBank in international database. When we obtain a genome data, we get different types of formats. It depends on the equipment which was used for genotyping. And often we face with FASTA, QSEC, FASTQ, SAM, BAM, GVF, GFF3, and VCF formats. and each format is quite easily integrated with the program algorithm while we interpreting genome. I think it's quite a lot of theory for right now, so let's switch to more viable, more everyday items. And what is our purpose? We need to get a useful interpretation of our genome, right? So we have to develop the environment which helps this biotechnology company thrive. And to do it, we have to predict sustainable business models and organizing business processes in these companies. It doesn't depend which country is it, Russia, in Europe, or Iran, or any other country. So, first of all, we need to evaluate the potential usefulness of the genetic test. And it depends on the medical field, like endocrinology, surgery, infectious disease, or eye disease. After that, we have to make research and development, analyzing all the data we have at this time, like genome-wide association studies and alignments in the world. After that, we have to protect the algorithms we use to survive in the market, because it is prohibited to protect the DNA sequence. But all up at the DNA we can protect by PCT, patent treaty, and thus we get an algorithm that we can use interpreting our data, and that helps us to make this product unique. And after that, after creating a product in diagnosis, we have to position it in the market. So it is very important to locate the niche of the people who are needed to make this test. So, in real life, the genetic company from zero is very hard work. So usually it is the separate branch of existing business, like the second part of laboratory business, like blood analysis or any other biomaterial analysis. Or it is developed in academia, in universities. And that is, in my opinion, the most sustainable way of developing gene test systems. A couple of figures. In 2011, the market of biotechnology in DNA was about $8 billion USA. And this market is growing 15% annually. So this market is quite attractive for new investors and new kind of all supporting businesses. And in Russia, there are several companies, and each of them are in different fields, so the competitiveness of the market is quite low. I do not know exactly in US or Western Europe, but in Russia the competitiveness is quite low. So I think in Eastern countries there is also very huge potential for growth. So our centers in Russia are partly government, partly private equity. So DNA lab is the biggest one, this one. And DNA diagnostics, Genix, is the Russian branch of American company. DNA in Siberia, DNA Center of Molecular Diagnostics, our company, and Municipal Moscow Fund for DNA Research. These are the major market research companies of DNA diagnostics in Moscow. I was very happy to find the research in Iran concerning this field, and I would be very glad to meet Dr. Suri Elahi Yazdani and Naruhi Neyad. They are researching genes of glaucoma and cytochrome, myocilin, and some other genes which, when damaged, cause very severe glaucoma, the blindness disease, the disease leading to blindness. In Europe, there are lots of academia institutions which are the basis for private health companies. And it is a very famous Netherlands medical university basing in medical center basing in University at Raboud, and a very highly expert Franz Kremers is focused on detecting genes which can lead to blindness. Also, we have partnership with Estonian company, IranBiotech. This company's focus is microchip, microarray for DNA tests, and they have different departments, not only endocrinology, reproductive, oncomarkers, but also ophthalmic services for DNA diagnostics. There is also the Institute for Genetics in Cologne, and we have some partnerships with them also. In UK, it is London's King's College, and there are several US biotechnology companies which are influence market a lot. So we have to use, and we use it with great pleasure, some of their products, because they make trends in the market, and they create a new field and new level of understanding of the human genome. These are Amgen. They bought some time ago Decode, the company which interprets